Okay. Good afternoon, everybody. My name is Veneshley Samuels, and I'm a PhD candidate in the MMRU lab under the supervision of Prof. Digby Warner. And today I will be talking to you about my PhD project, which is a new look at an old target towards cryo-EM elucidation of bioaerosol-captured Mycobacterium tuberculosis. So, just to give you some background, my research is actually conducted as part of a larger collaborative project, which is based in Masipa, the Aerobiology Research Center, where we have a large team of people who's primarily focusing on studying the aerobiology of the disease causing pathogen Mycobacterium tuberculosis, which is responsible for TB disease. What do we know about TB? Well, TB is a global killer and it is of special relevance in South Africa, but it is also an airborne disease. And so MTB transmission is much more complicated than it seems because accumulating evidence suggests that people who are asymptomatic can transmit MTB. And that makes it difficult for us to identify and block transmission. Therefore, investigating airborne organisms is extremely difficult because there are significant challenges involved in capturing and characterizing these organisms. So, in an attempt to study and understand the aerobiology of MTB, our team developed this really advanced technological and analytical pipeline to screen XL breath using a sampling chamber that we call a respiratory aerosol sampling chamber, where we collect aerosols from TB infected patients. Once these aerosols are collected, they are stained with a cell wall probe called Trehalose. Trehalose is basically a fluorescent probe that gets incorporated into the micromembrane via the antigen 85 complex, and it also enables us to detect viable organisms. Now, as much as we are using fluorescent probe to look for viable organisms, and as much as it is enabling us to detect positive live MTB bacilli, the resolution afforded by a light microscope kind of restricts our ability to obtain a much more detailed understanding and much more detailed information on the biology, including the cellular and structural features of the organisms that we are detecting. And that makes it difficult for us to determine which properties enable successful transmission. So this is where my PhD project comes in. My PhD project aims to address this limitation using electron microscopy to obtain high resolution information on aerosol captured MTB. So the purpose of my project is to identify and characterize MTB bacilli that we capture from these infectious airborne droplets, bioaerosols that are released by TB patients. And the main question that we are asking is, do MTB bacilli in bioaerosols differ structurally in cell wall composition or bacillary or metabolic state? So as an entry point, I've developed this sample processing workflow, which kind of plugs into our existing MTB detection clinical pipeline, whereby individually stained organisms are located on carbon-coated electron microscopy grids that are then subsequently analyzed and imaged using cryo-electron microscopy. So to start off and to basically develop the pipeline, we used lab cultured Mycobacterium M. smegmatis that was stained with Trehalose. And as you can see, we were able to show using M. smegmatis that we can find and we can isolate individually Trehalose stained organisms using fluorescence microscopy and finding these organisms on carbon-coated electron microscopy grids. We were also able to show that we can co-localize these organisms. So co-localizing and actually stained organisms using cryo-electron microscopy. And our cryo-electron microscopy data revealed some of the major cell wall layers, such as the mycobacterial outer membrane, as well as the peptidoglycan and arabinogalactan layer. So from here, we also tried to see if we could improve our signal to noise ratio. So we averaged cryo-electron micrographs. This basically just further resolved the layers that we were seeing that were initially visible to us, which is the outer membrane and your peptidoglycan slash arabinogalactan layer. That's what you see here in the intensity plot, where it's just further validated by measuring the distance between the two layers. Now, these results, it was very encouraging for us to see that developing this pipeline, that we can actually isolate individual organisms and we can co-localize them. So we kind of wanted to take it a step further and see whether we can do this on an actual clinical sample. So in this slide, what you are looking at is the results of an actual confirmed TB case where we collected a bioaerosol sample from a patient that was gene expert sputum positive. And this sample was taken before the patient started treatment. As you can see, well, I think it's important to mention this, but this is like our results is very much preliminary, but it is also our first cryo-EM insight on a confirmed TB bioaerosol sample. So as you can see, we are able to locate these stained organisms using fluorescence microscopy. We can also co-localize these organisms using cryo-electron microscopy. This made us very exciting because further encouraged us, and it also further supports exploring and studying airborne TB or exhaled breath closer to its native state. Some of the further questions that we have, number one is specificity. That is basically because the fluorescent probe that we are using doesn't just stain MTB alone. And then secondly, infectiousness. Well, with infectiousness, are the bacilli infectious or not? This is something that we can't answer for sure. We do know that they seem to be metabolically active enough to take up the probe. So we would assume that they are viable. So we would assume that they are infectious, but then again, we can't say. And then lastly, with the cryo-EM resolution. So I've mentioned earlier in my previous slides that some of the major cell wall layers that we noticed in our images were the membrane as well as the peptidoglycan slash arabinogalactan layer. So the existing literature reports on actually more than two layers, so 3 or 4 layers, depending on the technique that you're doing. And that is most important because the bacteria that we're working with, these organisms, they are extremely large. I mean, they are up to 500 nanometers in thickness. So you can only see that much if you want to get to the finer details. So we are hoping to move towards cryofib and cryo-electron tomography. As I mentioned, this is a large collaborative project. And this final slide actually just shows it all. We have the MMRU team, we have the Aerobiology team, we have the EMU, all the supervisors that's working really hard on this project to push it and to make it work. This actually shows you that the pipeline that we've developed, it was not just that summary that you've seen. Each and every step in that pipeline had to be optimized in each and every one of these locations where you find all these different teams. And with that, I just want to say thank you for everybody, as well as all of my supervisors for all of the support.
Thank you very much, Veneshley. We're back on time, which is a big surprise to me. Okay, so anyway, firstly, let's ask people to give some support and questions to Veneshley. Okay.
Thanks. Hi, Veneshley. Thanks. Great talk. Just a question. I'm not sure of the answer to this, but if you It must be quite hard to identify them on the grids that you